National 5 biology students. Let's do this past paper question together. So this one says a study was carried out to investigate the effect of using the chemicals ethanol and methanol on bacterial growth. Controls without either chemical were also set up. The results in the table showed the growth of 3 species of bacteria as a percentage of the overall bacterial growth in the control experiment. Okay, that makes sense. So we've got the 3 different species of bacteria. We've got their bacterial growth when they had the ethanol treatment. We've got their bacterial growth when they had the methanol treatment as well. And that is a percentage of the control, i.e. when they grew them without either chemical, okay? So if I was to sort of try and get my head around this, I would say control there, right? So no methanol, no ethanol in the control. They would all be 100%, okay? And these are proportional to that 100%. Okay, that can be a tricky thing to get your head around, but that's the way to think about it. Now, on the grid, it says complete the vertical axes by adding a label and a scale and plot the remaining bars to show the bacterial growth for both V. cholerae and S. paratifi. I think that's how you say it anyway. Okay, so we've got the ethanol treatment, which is the diagonal lines there, and we've got the methanol treatment, which is not get any lines in it. So what we have to do is we have to look at the V. cholerae one and the paratifi one, okay? So this one here, we're doing the ethanol treatment first, okay? So this one here is going to be 45, okay? So we need to do our first bar. Now, these bars are 4 wide, okay? So we need to do our first thing up to 45. Now, actually, we need to do the scale first, don't we, before we think about the bars. And what this is a representation of is the bacterial growth. And we just get this from the table. You don't have to work this out, okay? You just copy what's up here as a percentage of the control. Okay, perfect. And then what we have to do is find out the values. Now, before actually I plot my values, I'm going to just compare this to the original one. Now, if that's 36 for ethanol, if that's 36 there, that suggests that would be 30 and that would be 40, okay? So that helps me with my scale. This one here is 54. So that suggests just above 50 there. So that's going to be 50 there and that's going to be 60. Now, if we see that already from the scale, we can hopefully work backwards and do the rest of the scale there as well, okay? So the V. cholerae is going to be 45. So 45 is going to be round about here, okay? And then we go 4 boxes along, 1, 2, 3, 4. And the methanol treatment was actually 44, so that's just slightly below that. 4 boxes along, okay? And what we would do, you wouldn't do this, what Mr. White's doing, you would use a ruler so that we can keep those lines nice and straight, okay? And we would fill in our bars like that. Now we know that the methanol has nothing in it, but the ethanol has these diagonal lines, so I just put some diagonal lines in here just to indicate to the marker that this is ethanol, okay? And then we go to paratifi, which is 4 for the ethanol treatment, so that's going to be what, 4, which is going to be there. And the other one is 12 there, okay, so that's just going to be just above 10, isn't it? Okay, and again, we just do our lines down just to get our bars filled in. And again, we go like that. Okay, so your graph would look something like this one here when you finish drawing it. And that would get you 2 marks if you did that, and you obviously did your lines with a ruler, unlike Mr. White. Okay. On to part B. It was concluded that ethanol was much more effective than methanol at preventing bacterial growth. Tick 1 box to identify whether this conclusion is valid or invalid, okay? So was ethanol more effective than methanol? Well, let's have a look. When ethanol was used, there was much less bacterial growth. So that suggests that it was more effective in this first one. In the second one, however, the ethanol treatment actually has more bacterial growth than the methanol treatment, okay? So actually, ethanol is less effective in this one. And then it becomes more effective again in the last one. But we would have to say that this conclusion is invalid because it's not supported by all of the evidence. It's only supported by the evidence of 2 of the species of bacteria, but it's not supported, or it's not true, for the... Now, let me just double check this spelling here, the cholerae one, the V. cholerae, okay? So it's not true for the V. cholerae. All right, or you could go into more detail and explain exactly what it is, i.e. the ethanol was more effective at preventing growth for the first and third type of bacteria, or it was more effective at the methanol was more effective at preventing growth in the V. cholerae, or something similar if you want to be more specific, but that would be enough to get you the answer there. Okay, see, the researchers used a 6% ethanol solution in their study, predict the effect of using a 3% ethanol instead on the growth of the bacteria. Okay. So it's a 6% that causes this. What would happen if you halved the percentage of ethanol? Well, in that case, you would inhibit bacterial growth less, which means that more bacteria would grow, okay? Because remember, the job of the ethanol is to reduce bacterial growth. So if you make it less concentrated, more bacteria will grow, okay? Or bacterial growth, more bacterial growth is what I probably should say for this one. Okay, there would be more bacterial growth, or you could say bacterial growth increases. Okay, something like that would be acceptable. All right, folks, hope that helps. I'll see you in the next one.